Freehand.
So we're going to copy that side. We're going to use this side. We're going to bind to this to copy that. Okay? The only difference is T's are now used. Okay? So if this is A, C, G, T, A, what is my RNA strand? A, C, G, U, A. Because what are we doing? Copying it. All we do is switch the T's to U. That is my RNA. Okay, that is the process of transcription. We're going to do the second half of today and all of that. Okay? That is transcription. Now, what we need to do is we have our RNA. Okay? So, it's a whole bunch of nucleotides. And let's just say it's A, C, G, A, G, A, U, G, A, C, A, G, C. So, okay? What end is this?
A, C, G,
5 prime. This therefore is
3 prime.
Okay, so we have center orientation. What comes in and binds to your mRNA? So let's just list this out as mRNA. What comes and binds? Small subunit. Excellent. Small subunit is going to come in and it's going to bind to our RBS. Ribosome binding site. Okay, and then it's going to move along until it gets to what? AUG. Once it hits here, another subunit is going to come in. That's the large subunit. And we're going to recruit what? tRNAs. Excellent. So, what does AUG stand for?
Matt.
Yeah, it's the start codon, right?
Yep.
Okay. So, MET is going to come in. What is the anti-codon to the codon? U, A, C. Because U binds to A binds to U, C binds to G. Excellent. That's going to come in and bind here. Then as this moves along, we build a polypeptide chain. So, AUG, AAA, ACA, AGC. That will build your polypeptide chain. This is my N termini. This is my
C terminal.
If we draw out MET, there's MET. MET is going to come in and it will start this polypeptide chain. So now this is my N, C, and we're going to build that. This is the process of translation.
Or translation.
Okay? So that is what we did on Monday. So now we should be finishing up page 2 of the worksheet, and we're going to be able to continue on, because now we need to do elongation, right? We've started the process. Now we have to finish elongation, and we're going to do releasing. So we're going to figure out how this all comes together to form a polypeptide chain. This process is building what structural level? Primary.
Primary.
Because I'm establishing my peptide structure, right? Each one of these lines represents a peptide bond that will be made. It'll come in and will form a primary structure. Once it gets released, secondary structure and tertiary structure will happen automatically because of the chemistry. And if it's quaternary structure, we get assembled in what organelle? Rough ER.
Rough endoplasmic reticulum.
Okay? Let's finish up this question. I was doing this to like crazy. Okay, let's talk to each other. Our students are great on this one.
I got cell wall mitochondria.
Cell wall. Mitochondria?
Yes. Those are not in eukaryote.
Oh, okay. In eukaryote or in prokaryote?
Prokaryotes,
Yeah.
I meant. Prokaryote.
What kind of chromosomes do you have? Linear chromosomes. What kind of chromosomes does a prokaryote have? Circular. So let's have that. Do they have proteins? Sure hope so, because they're doing function, right? So therefore, if they have proteins, what must be needed to make them?
Ribosomes.
Ah, so they got that. So that just eliminated like a quarter of us, okay. So then the question becomes, do they have a cell wall and mitochondria? What is a mitochondria? A prokaryotic organism. So they do not have mitochondria because they are mitochondria. So that was incorrect. Do they have cell walls? Yes, they do. Are not found. So the mitochondria is the only thing.
Oh,
I was wrong on the cell walls, but yes.
Yay!
cell wall?
Let's keep going.
Karyote.
I was pretty sure cell wall.
Cell. completion factor, release factor. Okay.
This is a release factor.
Release factor.
This question? Oh, it was just asking. Oh. Yeah, it's listening to animals. Wait, no, the animals are things. Oh, I would think that was, I mean, Oh, yeah. Or like why? Yeah, yeah. Questions are always those cracks. So I don't know the actual like. Yeah, I just got some pressure or whatever. And I just sucked and I just walked it out. And it was
Am I crazy? Like, I thought that's I thought it was a big fan to be
Stop.
Like it's like so
Wait, for which one?
You wouldn't get it the whole month. Correct. It would still face tear.
It would keep going?
It'd keep going. Continue elongation.
I know I like crossed it out.
Oh, I have to go right now.
Okay, but I thought like, what do you mean to take the part? I know, right?
There's several stock codes, but yeah.
In this case, it's
I don't know. But that one.
new ID,
Where is that?
Maybe.
like the biochip.
Seven is something.
UAC.
Your code on the label.
Is it AEG? Is AEG is
Well, okay, so if your exams are supposed to like
Wait, TRNA scan.
I feel like I feel like it's so obvious. Oh my god, I'm literally actually No, I definitely did.
What's tRNA stand for?
Transfer RNA. That's what tRNA stands for. AGG. UAG. UGA stop.
Oh, I see. I thought it
UAG stops. UAG. It's a release factor and enzyme.
Yeah, it's just necessary for certification.
It's a release factor.
How do you know? How can you know things like
The release factor on enzyme. This is the release factor, right?
It doesn't. The release factor.
Protein.
Yeah, the
Which
release.
a protein is made out, but it's not an enzyme.
Yeah, release factors are proteins.
And then like I'm doing something and I'm like, that is scribbled up. What exactly is that? I would say yes, right? 'Cause that's what yeah, that's true. Please help. Yeah, it's now.
Oh, my bad.
Oh, we just
It's okay. Stay longer.
yeah, it's the last one, but
Is this one?
yeah, that's the reason.
Oh, Dodgers.
Yeah, yeah. That's what I was thinking. Figure it out.
Yeah, so it's catalyzing a reaction, so thereby diffusion. All right, a couple questions for you all. Okay. So I should just.
I put A. What is
I got C.
C?
I did,
Yeah.
But he said that quaternary was not until
Quaternary is quaternary
the
is multiple polypeptide.
rough
bonds.
ER. Does that have to do anything? I don't know.
Probably might not have something to do with H.
I was between A and B, but I put A.
I think.
So, we have our mRNA. It runs 5' to 3'. This is a snippet of said RNA. This RNA is probably past the wall out to the building in length, okay? If we're to scale it up. It is really long. We're building tons and tons of amino acids off of that. Most genes are 500 to 50,000 amino acids long, okay? So, this is going to be really long. This is our ribosome binding site. It's going to bind to this complementary, okay? So that means the ribosome also has what into it? Has nucleotides, right? Can bind. So a ribosome has nucleotides as well as protein. It's that combination there. All right, so this comes in and binds initiation. Then it moves along until it finds what? Start codon.
Start codon.
That tRNA comes in. When we look at tRNAs, tRNAs are quaternary structure with RNA as well. It's going to have your anticodon. So if the codon is AUG, what is the anticodon?
AGC? No.
UAC.
UAC. Okay.
Right? AUG, A binds to U.
U.
G binds to C. There we go.
C. Okay.
So tRNA is called tRNA because it's transferring and it has this funny T shape. Coincidence. Has a 5', has a 3'. That 3' end is sticky. Therefore, we can do something with it. We're going to stick an amino acid onto it. Okay, this one has alanine. That means there's N terminized there, C terminized there. If it does not have an amino acid attached to it, this is often called a release factor. So it'll come in, bind to the mRNA like normal. The ribosome will move. However, was there any continuation off that polypeptide chain?
No.
No, so the polypeptide will now float away, and the RNA will fall apart because it can't build on something that's not there. That's why it's a release factor. Okay, so it'll come in and bind. Once we have met, we're going to set our reading frame. AUG, so the next one is GAA, then so forth, in perpetuity. This is our N termini, this is our C termini. Each tRNA is going to come in, bind, and release the amino acid. And we're going to release it and keep going and keep going and keep going. Why, then, is it important that this is held together with hydrogen bonds? Because this is hydrogen bonding here. This is then going to hydrogen bond there. Why is it important that it's hydrogen bonding? So it is weak. It's weak, so it can move along and it can fall apart. If it was strong, what would happen? Nothing. It wouldn't work.
Oh.
Because it wouldn't move, it wouldn't be able to fall apart, it'd be a one-time thing, and it just wouldn't work. So the fact that it's hydrogen bonding allows it to be really weak, so therefore it can move, it can slide.
So D?
Yeah, D.
Whatever it is, yeah. Weak.
Weak.
Alright? Next question. Closing up in just a second. A couple people don't believe this stuff.
I still don't believe you. Are you sure?
AGG, UGA, AUG.
Circle, stop. Stop codon, UAG.
UAC, UAG,
Down. UAC. I don't see it. Oh yeah. Well, that was pretty close, wasn't it? AUG. It's UAC, right? not G, UAC.
It's UAG, UAA, or UGA.
Oh, not UAC?
Alright, 10 seconds.
I thought it was UAC.
Alright, Dr. Stern.
It looks like it's just PR. If you go by threes after AUG, it's like there's three and then like there's already. So I think it's going. I don't know.
So what did you do? AU?
AUG, start codon, and then UAG, stop codon.
UAG.
Yeah, I did that too.
What is the start codon?
AUG.
So we go along. What's going to be over here? Ribosome binding site. We move 5' to 3'. So we go along until we reach
AUG.
AUG. Okay, there's our start codon. Then we go every 3 nucleotides then out until we reach what?
UAG.
That's a stop codon. There are multiple stop codons, but that is just one of them. So in this case, this amino acid strain is very short. It's possible that exists in nature, but it's not. This is more of an example, right? Typically, you'd be 3,000 base pairs that way. Often, stop codons also come in repeated sequences, so you'll find like 3 or 4 stop codons in a row. Okay. Yeah.
If the RPS is varied between like species and stuff, how are you supposed to identify that?
Typically, it's going to have that repeating segment of A's and G's. Okay? It's always before the start codon. You have to guess. This is that repeating segment. I'm typically never going to ask you to, pinpoint it exactly. I'm going to be like, where is it? Should it be? Okay. All right. Codon tables. For those that haven't seen those before, you just start with, like, if I want A, C, G. So we start with A, then we go to C, and then you find the G. That's theory. UAC is
Tier.
styrene or whatever it is, yeah.
UAC?
Okay? So that's how they work.
So you find the first letter by looking at these ones. So you pick
A.
U. Yeah, so if you pick A, these ones all start with A. And then you go for your second letter, so up here,
U.
so it's AU.
Uh-huh.
So all the ones in here are the AU. All the ones in here are AC. All the ones in here are AA. All the ones in here are AG. And then for your third one, you look here. So if you want AAG, you go A. For this one, so AAG is going to be there. If you want ACU, you go A. You go to C, and then you find the U, which is just going to be at the top.
Oh, here.
Yeah.
Oh, got you. Okay, thank you.
No. Oh, you mean, so those are different questions. Yeah. So yes, the release factors.
Oh my God, this is a lot.
Ribosome is moving along the RNA, but there's nothing holding the. It's just gonna let
Oh yeah. So that's not the right Why would these be there? Recording or what? Why would these be small? I just like, I don't get it. I don't know if it's like.
AEG is the codon for MET, amino acid. The good thing about the MET, just don't worry. MET is the amino acid. So amino acids are serine, methionine, whatever amino acid that is, that is corresponds to as long as it corresponds to the so MET only exists once, but ilanine, AU, has a different amino acid, different got the same amino acids, codons. No, there's no codon. So the next one is an AAA, GAA. So tell me what codon that is. Okay, that's the next. So another TRA is going to come in, and it's going to be GLA.
Maybe that's the answer. I'm reading it right now.
That's such a bad idea to have this on.
Correct, just as it did.
With the TRC.
That's correct, just as it did. Okay? So they extended on, that's elongation. And then it terminates when the release factor.
A release factor is
Did check that right in the first place.
But not the,
There's no release factor. Release factors are tRNA with no amino acid.
yeah, because it.
It releases it. It just makes it fall apart. So if I'm moving along, okay, my ribosome is moving along. Here's my polypeptide chain. If I have a tRNA comes in, this is going to move. But if it doesn't have an amino acid, what happens to this thing? This floats away. Then therefore all this just falls apart.
I don't know what's on the TRNA.
Yeah, so what's different about this than these ones? What does it not have? Correct. So the codon is different, and the amino acid is not.
Which diagram is it?
Yeah, so it's peptide.
Yeah, right? Like, to the right?
You know how like
Yeah, yeah.
the second one is this cotton.
Yeah, that's what I was
The name of the process, no, but like literally.
It's like the Yeah.
But this might be like manipulative games, but it makes you like read like the same questions over and over.
Did you write
Yeah.
FMAT, tRNA, mRNA, ribosome, and anti-codon.
Now you done?
No, I got stuck on a question.
I haven't gotten to that yet.
What happens on the last side is the RNA is moving. This is going to want to move. It's not going to
This is
It will
translation.
bind, the tRNA is able to bind to the
Translation.
mRNA. It's not able to bind.
Ribosome.
So what happened? Where did the mRNA, disappears, right? It goes away. And then what happens to the ribosome? Falls apart. What's a release factor? tRNA without an amino acid. So the ribosome is moving along that mRNA. It has a tRNA. That tRNA comes in and it binds to the mRNA. However, is the polypeptide chain continue to be elongated? No. Therefore, the polypeptide chain does what? Goes away. Goes into the environment. Okay? And so the mRNA is continuing to move. The ribosome thinks it did its job. However, the amino acid chain just falls away. Can I build anything from something that's not there?
No.
No. Therefore, it all just falls apart, and the enzyme is ready to be reused. So it could go back to AUG and start over again, or goes away. Doesn't matter. All right?
I'm a bit confused what I'm supposed to be doing for 6.
Okay. I went in order. Being less pain in the ass.
Cell. So what are we confused with?
I just don't know what I'm supposed to be like filling in for these blanks.
In many different types of proteins, there's an amino acid threonine where an alanine should be. What's the problem? What's broken?
Primary structure.
No. So if you have many proteins, means is it the mRNA that's broken? Because it's in many different proteins, right? So this has to be a broken tRNA. If there was anyone else.
Why? Yeah, it was me, okay? Not her.
They're so funny.
What is the next head up? Will be read by the ribosome in the schematic above. Triplet nature codons. Covalent bond between which two molecules keeps the growing protein chain attached to the ribosome? H bonds. Which two molecules? Which direction will the ribosome move in the model shown above? 5 to 3. Come on. Roll tRNA.
That's like, you can take course, right? Because that's like it's. What would it be?
Yeah, that's
Yeah, that's a. Yeah.
Yeah, there's no intent.
What is the purpose of translation?
Convert genetic information into proteins.
All right, guys, let's come back together. What is the purpose of translation? To make proteins. That is it. To make a protein. So we take mRNA and we're going to make a protein. What enzyme are we using? Ribosome. We use tRNAs and ribosomes. How are tRNAs made? Talk to Shark. How are tRNAs made? How are tRNAs made? With enzymes. Excellent.
Ooh.
They are
Enzymes.
correspond to a gene and an enzyme. Okay, so you make tRNAs constantly. Another gene and another enzyme is responsible for the manufacturing of tRNAs. So, let's imagine you have a single protein that has an error in it. A single protein. Where do you think the likelihood of, what caused the error? A single protein has an error in it. What's the cause of that error? Probably the ribosome made the mistake. Okay? Because it's a single protein, single error. Now, we can look at the mRNA. If the mRNA is correct, and it's a single protein, single error, it's most likely the ribosome. Now, let's imagine many proteins, many different proteins, all have the same error in them. It's probably the tRNAs. So it might have been the enzyme that made the tRNAs is the problem. Because many different proteins. If it was just one protein, okay, we can isolate that down to one subunit, one issue. But if it's many proteins, it has to be the thing that made the tRNAs. So errors can occur in this process. Errors are very rare. Okay? It's just literally chemistry. Makes things very rare. A G does not want to bond to another G. G does not want to bond to A. It's the wrong stereochemistry. It's the wrong shape, wrong size, wrong polarity. It's just not going to happen. So it is very, very rare that those things can happen. But let's say it does. Well, is it going to occur in many proteins or just the one? Probably the one. Now, let's imagine you had a mutation early on in your lifetime. Okay, so when you were still an egg, there's a mutation. That could have changed your genotype. It's a new allele. New alleles might make for a new polypeptide chain. Let's imagine on the 27th codon, you have a mutation. You went from CAC to CAU. What has happened? You went from your wild type is CAC. Your mutant allele is CAU. What has happened?
A mutation.
What's happened?
I think a mutation.
Yeah, they are.
All right. Your wild type is CAC, which codes for what amino acid? Histine. The mutant is CAU, which corresponds to what amino acid? histine. Therefore,
It'll not change.
no change in phenotype. This is a silent mutation. Okay? Mutations can be silent or they can show up. They can be beneficial or deleterious. Let's imagine the wild type is still CAC. But now we changed it to CAG. What has happened?
Then you'd
Different
have,
amino acid.
Then you'd have a phenotype.
This is a substitution. We've changed amino acids. Now as has happened. That will show up in many proteins because you have the allele for that. Okay, this isn't a translational error. This is a genetic error. Let's imagine the wild type is CAC. Yours, the second C got deleted. What has happened? This is a frame shift change. A frame shift. So we went from C or whatever, we're going to go with this one. CAU, okay? So there's our AUG. Okay, we're going to go here. GCA. This A gets deleted. Now all the other amino acid sequence is out of frame. So will you produce anything similar to the original protein? No. That's a frame shift. Frame shifts are crazy when they happen because it changes everything. Okay? Let's imagine you have an amino acid, UAC, and now it gets coded for UAA. What happens?
Stops early.
It stops early. So this will be what's called truncated. You truncate the protein. You've shortened the length of the protein. That's often a very big problem because now your protein, instead of 27,000 amino acids long, is 17. Is it the same thing?
No.
No. There we go. That is a truncated protein. Okay? Let's keep going for a couple more minutes. Let's finish this up.
The rate of initiation. Well, if the rate of formation increases, then the rate of initiation. Oh, okay. Yeah.
This is sugar. Yeah. No, it's not.
Not yet.
This is a large ribosome subunit.
Right.
Well, this one's saying the affinity of a large ribosome subunit per small ribosome. So the connection between the two.
Yeah, I think it came out kind of good.
Oh. This one's asking. Yeah.
Join the class. And then somebody is going That's what I did. Red mine. Oh, and I showed him these questions. He creates like
So is this like the tRNA or the phosphate or what?
So that's,
Structure of the release factor.
yeah, that's the tRNA. So it's the
Is that include the phosphate in there, or am I not seeing that we don't have it? Because this is the amino acid.
So the tRNA includes the phosphate because the tRNA is just the chain of ribose and phosphate.
You're all right.
Yeah, no, I need a
It's like that thing that you showed like here. Are you done?
I think so.
Okay, let me ask you then about this. Every time I want to ask you.
Every
I
time he
think he
asks
does
a
it on
question,
purpose.
he asks a question.
Okay, 10 seconds.
I put, oh. I
put them all.
All of them? I did A, B, and C.
I just did A, B, and D.
A, B, and C I did.
I said they were all true because
No protein product?
if I like depending on what they mean by change, it could just be a change in the amino or a removal of the base, which could result in all of them, or a change can just result in which amino acid is produced, or no change. Or if it's removed, it could be an early release.
Your wild type is CAC. It's been changed to CAU. This results in
Nothing.
Crazy changes.
no change. So 2 is correct. CAC is the wild type, and it changes to CAA. That results in
Truncated protein.
CAA? No.
No.
Single amino acid changes the protein. So 1 is true.
Oh, good. 1 and 2.
You have
Wait, what?
UAU, and now it's UAA. It is a truncated. So 3 is also true. You go from UAA to UAC, and now it is longer. There we go. 4 is also true. You go from AUG to AUA.
Never starts reading.
Never starts.
You got
All
it.
are true.
You got it.
Ooh, okay. You got it. For termination integration questions number 4, what did you say? I said due to the absence of the amino acid, it can
Wait, which
only
one?
tear apart the ribosome.
Oh, how does it... Number 4?
Yeah, number 4 on termination integration.
I just did structure equals function. I wasn't too sure about that one actually.
All right. Let's do this. What do I start with?
AG.
That is
Met.
met. Therefore, this is my and termini, and we're going to build on the C. Somebody tell me what GCA is. Alanine.
G, C,
Then we got UAC.
alanine. U,
Tyrene.
Firing.
I think. No, TYR. Never mind, I'm way off. Too many of them.
Let me see if they know how to.
Okay. TYR, you get this. Then we got AGC. Cyrenine, yes. Oh. You know what? My pen is messed up. Oh, that's good. So we got MET. GCA was alanine. And then UAC was
Firing.
TYR. That one was SEER. And then CCC,
Proline.
UAG, stop. So, there's my N termini, that is my
C termini.
So when you look at this, it'd be N, C, C, N, C, C, N, C, C, N. I have this one memorized. There we go. So this is primary structure, and to C termini. This would be your main structure, right? That's what it would come out to look like. So whatever these R group interactions are. And then they would come together and start forming secondary, tertiary, and eventually probably quaternary structure if we have multiple polypeptide chains. Okay?
Mm-hmm.
So, if I give you a DNA sequence you should be able to transcribe it, then translate it to produce your polypeptide chain. We're going to do transcription here in a bit.
Which is a protein.
So let's reverse this. Let's reverse engineer. Okay, so we've done this, so everybody knows this. This is my mRNA. Somebody tell me what the coding strand will be for this mRNA.
The reverse.
Talk to her. Come on, what's the coding?
Is it the reverse of the?
What's the first three nucleotides? AGG. 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 And then it is T. Because mRNA equals coding. T becomes U. So all I'm doing is reversing this, U to T. My template strand, so that's coding. This is my template, would be complementary. T, C, C, T, C, C, A. Okay, so we can go forward and reverse.
Is the coding strand from the RNA polymers and the template is from the DNA?
No. This is just the opposite strand to what the RNA polymerase is binding to. So, in reality, what this looks like, right? So this is 3', this is 5, this is our template. This therefore is coding. My RNA is going to be made from this. So my RNA binds to the bottom, which duplicates the top.
Okay.
Okay, because we build on the 3' end. This is 3'. This is 5. So by rule, can I even build off that? No, because I have to have a 3' end.
Yep.
So I can only build complementary to the 3 to 5. Okay, so great question, because this would be that's your mRNA. This is DNA. Wow. And this is DNA. Keep going. Let's finish this up. Then we'll start on the next thing in about 5 minutes.
Oh my God. Okay, let's go here. He was asking about number 3. Did you already? Oh, you know, number 4. And I was well, do you have these ones here?
Yes.
I didn't do the true and false ones on that one because I was pretty much confused.
So I said the mRNA function is canary's genetic information from DNA. And I said it was necessary for initiation.
Yes.
Necessary for termination.
Oh, really? You
Yeah,
think so?
because if we don't have the mRNA, the tRNA can't read it.
Okay.
And then for FMET, I said that was canary's amino acid to bind to start codon.
Carries the amino acid.
Yeah, and that, the FMET, I believe, is the specific one that is for the start codon, the AUG.
Yeah. But to bind with what?
Carries amino acid to bind with start codon,
Oh.
so the AUG. I said that was necessary for initiation and necessary for elongation, but not necessary for termination.
I have it. What?
I said it was necessary for initiation because it's the first one.
Okay. I don't know at what point was initiation. That's what I just thought AUG. That's why. But
I figured like
Like,
initiation
it's right-
included the building of
an
the
AUG.
ribosome and the start.
you're
of
right
the
because
codon.
it's right here, right?
I mean, I don't,
So would the anticodon be started?
so,
I'll ask them.
so this is the anticodon, the
Yeah.
UAG.
Yeah.
And so that's going to bond to the
But
AUG.
is this initiation, would you think that's initiation?
I think initiation is just kind of like everything that happens right,
Right
before
here.
it starts coding.
Right
Yeah.
here.
The initiation would
From
include
here to here.
I think it would include, the building of the ribosome around
Oh,
the
okay.
mRNA and the starting of coding.
Gotcha.
So that would include RBS like binding to the ribosome, the tRNA binding to AUG, and then the large subunit like attaching to the top. I think that's all initiation.
Yeah.
And then here for platform, you have yes, yes, no. Yes, yes, yes. You have yes, yes.
Yeah, I have yes, yes, yes as well.
And
Yeah.
for a release factory.
When read stop codon releases all components.
Wait, didn't he say that for release factor? No, never mind. What did you say?
I said when read stop codon releases all components because it releases the protein chain, it releases the top, it releases the tRNA, and it releases it just like everything goes away.
Okay. Yeah, everything falls apart.
Everything goes apart, and then I said
Stop codon.
I said no, yes.
I did that. Yeah, I said no, yes.
No, oh, I got that one right.
And then
Anticodon?
Anticodon, I said binds to the codon of mRNA.
And then you have no? No, you have yes, yes, no. Backwards.
I think that one was I think that one could actually be yes, yes, yes. Because it is necessary for you need an anticodon. So I think it's actually yes, yes, yes.
Yeah. Termination?
Yeah, you need the
Maybe not the
anticodon
last one.
to stop it.
Well, because you need the anticodon that reads the stop codon. So I think it actually is yes, yes, yes.
Yeah.
I think I was confused at that point because I didn't know that release factor had an anticodon.
And then for one, several ones? Oh, terminate. Oh, you have to put termination.
This one's true. So imagine you place
That's
4 purines with clonidines and ribosome binding site. I'm not sure. I wasn't sure about the first one.
Well, that's
That was the one I just wasn't sure about. The affinity of small ribosomes would decrease. Because if you replace the purines with pyrimidines, those are the ones with weaker H bonds. So the H bond that connects to the so the RBS will then have a weaker bond to the ribosome, so it'll attach less and will not be as strong as a bond.
we can ask him.
Yeah.
How do you get the next codon that will be read by the ribosome in schematic? Oh, in this schematic.
Yeah. So it's
So
just
GAA.
yeah.
Okay. Okay. Stop and reflect. This is just reflection, right?
Yeah. Just make sure you understand how is the reading frame established.
Okay. What about in this one? What do you have for the reading? Oh, let's see if we have the same one. Peptide bond.
So a covalent bond between which two molecules?
We're in your. Okay, so we got peptide bond. Do you have peptide?
I said tRNA and amino acid.
Okay, we got that from him, so this is pretty accurate.
Peptide bond?
Oh wait, yeah.
Alright.
I think that's for the elongation.
Wait.
Yeah.
Do you have it different?
I said tRNA and amino acid.
Oh yeah, elongation integration. I was confused on that one. I was thinking H bond, but
Never mind. This is not that I got it from him. I just
peptide bond.
Okay. Anyway, which direction? To the right.
I said towards the 3' of the mRNA.
I said 5 prime with 3
Oh, that's more scientific.
the 3'.
Termination what?
I said towards the 3'.
Towards? Oh, towards 3 prime.
Yeah. It's gonna go towards the building cell.
Of mRNA. And then what is the function of tRNA?
I said carries amino acid and anticodon, but that's what that is. Like, it's the translation.
So translate and carries an anticodon. And,
New bond.
and then for this you have B, you have A.
What would happen if all the tRNAs in a cell with anticodons for glycine carry glycine amino acids instead?
Rick,
So, in many proteins, the same problem is occurring. An amino acid has been switched in many proteins. Therefore, what has to be the cause? tRNA. Because a tRNA is in many different proteins. tRNAs are made by their own genes. So if the tRNA is broken, that's going to show up in many proteins. If it's a single mutation showing up in one protein, that's a mistake in that protein. But if it's showing up in many proteins, it's a mistake somewhere else, like tRNAs. Okay? Keep going.
okay, so we were here. A covalent bond between which two molecules keeps the growing protein chain attached to the ribosome. We have tRNA and amino acid, or peptide bond.
So, a covalent bond between two molecules keeps the growing peptide chain in the amino ribosome. It is a peptide bond.
Absolutely.
And the reason why is if the tRNA that comes in and it is not forming a peptide bond, what happens?
It's gonna break apart.
It breaks apart.
Breaks apart. And then here we also have what could happen if all tRNAs in a cell with anticodon for lysine carried glycine amino acids instead of lysine amino acids? Translation would still occur?
Correct. So if it's just carrying something incorrectly, it will still occur.
So not stop.
That is a tRNA problem, not a.
And last but not least, can I ask you this last but not least? Because we have different ones. And we don't know where like it starts and so. Okay, so mRNA carries the genetic information necessary for initiation, yes. Elongation, yes. Termination, yes. Yes. Yes, no.
Sure. Okay.
Yes, yes, yes.
Yes.
No, no, yes.
Correct.
Yes, yes, yes.
Yes.
Okay.
Does it matter? Like, or are we correct? Or
That is correct. You had it.
Ooh, yay.
good job.
I was wrong with the last one.
Oh, we
Me
forgot
too.
to ask about
Me
this
too.
one.
I was wrong with the last one.
So I was right with the last one.
Yes.
Ooh, good job, Mar.
Last but not least. Just one more question.
Just one more question and then we're almost. This stuff is so hard to understand.
So yeah, it is correct.
So it's true,
Yeah.
false.
Oh, true, false. Okay.
Nice. So that this is supposed to be peptide bond.
Keep asking, Mars. Give us all the information.
All right, let's move on. I have a new question open. New session, new question.
Which base will be added to the RNA string next? Alright, T to U.
So, I was just wondering what's a base. Is a base 3 letters? A base 3 letters? What's a base?
It can be either A, T, C,
5
G, or
seconds.
U.
So 3 letters.
Okay. Or something like that.
I talked to her.
I picked U because it's an opposite the T. What did you get? What did you guys get?
I didn't have a chance.
Okay.
I cannot explain. You say what?
I just say but it doesn't make sense.
I have no idea.
Yeah, I picked U.
Oh, there.
What process are we doing? What process are we doing? Transcription. This is transcription. Transcription is done by what enzyme? RNA polymerase. Nice job.
RNA
It moves what direction? 3' to
5.
5', because we build on which side? 3. We
prime.
always build on the 3
prime.
side. So therefore, are we moving this way or that way? We're moving this way.
Oh, to the left.
Because we build on
Okay.
which side?
On 3.
3'. This is the 3' in. This is 5. So we're building 3. So therefore, we're going to bind A to T.
T.
Can't be T because it's RNA,
RNA.
therefore it is U.
A to U. Okay.
The next code is U. Then who is
You
it?
had it, right? That's what you
C. Then G. Then
C.
G.
G.
Then C.
C. And then U.
U. Where is my
I'm not kidding.
where is my point of initiation?
AU.
Promoter. Point of initiation. This is in the reading. Talk to each other. What is a promoter? What is the point of initiation?
What's a promoter? I did the reading and there were like 4 quizzes.
Yeah.
And it just busted my brain, so I don't know anything.
I thought promoters were just in
What is the promoter? What is it?
I don't know exactly, but I know it helps to bind the.
Is a sequence that binds what? RNA polymerase, right? Binds your sigma on RNA polymerase. So it's a specific sequence that says go from here. I'm going to make my RNA at this location. So
Polymerase.
is a specific sequence. The point of initiation is this point right there. That's called your +1. So in lab this week, you're going to get sequences. Long strands of sequence. So if this down here is 3', this is 5. This is called +1. Okay? This is your point of initiation. This is your -10, -35. These are called the promoter. Okay, this is the initiation. And therefore, that just tells it where to bind. Notice that these are ATA and TATA boxes. Okay, ATA and TATAs. So therefore, this side must be what? Coding or template? Dr. Chair, coding or template? The bottom is coding or template? How do you know?
I think it's
Template?
It's template. T to A to T, T to A.
Wait,
C
I
to
can't
G,
remember.
A to T, C to G.
I can't remember.
It's template.
What's the difference between Tata and Nada? What was that? I remember
ATA and TATA are the same thing.
they're the same thing.
Yeah, ATA and TATA are basically the same.
Is there?
What direction
They're
is
going in-
RNA polymerase going? Right to left or left to right?
Right to left. Wait, what?
Left to.
Left to right.
It's
Okay.
moving 3' to.
5.
So therefore
it's-
we're going this direction. Therefore, is the top the strand coding or non-coding? Top is coding.
Coding.
Therefore, the bottom must be template.
Template.
In your group, tell me the first 5 nucleotides of the mRNA.
One is Matt.
That was it.
Oh no. That's not.
No, what they say. The first 5?
So does
Yeah,
it
the
start
first 5
with
nucleotides.
T? So it's going to be identical to the coding. So it's going to be AUA.
I am so lost.
Me too.
AUA.
What direction am I moving?
3 to
3
5.
to 3 to
That
5.
direction. So I'm building the top. So I'm copying the top. What is my saying? mRNA equals
T
coding.
is.
T's become U's. So what is my sequence? I start at the +1. So my sequence is
AU.
A, U, A,
U,
A, U,
G,
G,
A,
A, C,
C,
C, A, blah, blah,
A.
blah. Because I'm copying the top. Okay, that's my mRNA. That is 5' to 3'.
I still
I
don't.
build on the 3' side.
I
What
don't.
side do you build on? 3'. What direction does RNA polymerase go?
Left to right.
Never left to right, because what am I going to do to you guys? I'm going to reverse it.
So it's right to
Damn well
left.
believe on your exam it's getting reversed.
Oh, okay.
Okay? It's going to be reversed, it's going to be backwards, forwards, it's going to be every direction
Up and down.
possible. Because you have your orientation. -35 to -10, you're +1. What direction am I going in?
Left to right.
3 to 5. You go 3 to 5. Therefore, can I add anything from the bottom strand? Because what side do you build on? 3'. Therefore, you have to be copying the top by binding to the bottom. Just as I did here. I'm copying the top by binding to the bottom. Because I'm building on which side?
3.
3 to
prime.
5. A 3. Building up 3 primes.
What direction is my RNA polymerase going?
To the left.
This direction.
To the left.
To the 5. Always to the 5.
Okay? Now you figured it out. So if I know the direction, therefore this side must be,
3'.
this must be my template. So we're still copying this way, but now I'm just copying those ones.
Mm-hmm.
All right? Let's have more fun. Okay, so that's still 5.
Three to 5.
What direction am I going?
Left.
That side to
Three
this
to
side,
five.
right?
5.
RNA polymerase is going 3 to 5. Therefore, who am I copying?
The bottom.
The bottom now becomes our coding. The top is the template. So I can reverse directions. All right? Let's have real fun.
He's getting confused.
Tell me what is going on.
Is there an
Talk to
RNA?
Jerry. Tell me what's going on.
It still should go 3 to 5.
There's an RNA binded to the top and the bottom.
Huh?
There's an RNA polymerase binded to the top and one bonded to the bottom.
RNA polymerase binded to the top and bottom.
So one's copying the template and one's copying the coding strand. But both are coding and both are templates.
What is going on here?
There's an RNA
what do you
polymerase
say?
on both the top and bottom.
Excellent. Nice job. There's an RNA polymerase on both. We are transcribing 2 genes. There's a gene on the bottom and there's a gene on the top. We're transcribing. They're going in different directions. They're independent of each other. Do you think just one side of your whole chromosome is the only thing with genes? Hell no. Now let's be even crazier. How many genes do I now have?
40.
3 or 4 or 5. They can be running simultaneously in the same orientations. Because what if we have an AUG here that is out of frame to this AUG? Holy cow, now we have more and more and more. So this is where you can even get a single transcript with hundreds of gene products. So the chemistry is going to dictate. If I know where my +1 is in orientation to my promoter, -10, -35 stands for what?
Promoter.
Promoter, which is my ATA and
Tata
TATA
box.
boxes. If I know where my ATA and TATAs are, and my +1, I know my orientation. I know what direction RNA polymerase is going. If I know the direction of RNA polymerase, I can detail what is coding and what is template. Okay, so do you think I'm ever just going to be, I'm going to go over left or right just to be nice? No. Hell no. Because that's not how it works. In our minds, we like to orient things in certain directions. That is not how the real world works. These are in weird configurations. Remember, they're helices. We got to break them apart. We got to do the orientation. So it's all based on the polarity. So count your damn carbons.
Sumira said
Okay.
it yesterday.
Did she say it?
Yeah, she did.
Count your. She said the F word.
No.
She said, count your
I
F.
don't want to know.
Count
I
your
want to know what
She
you're
said
talking
F.
about, okay?
But she said F. She said F.
I'm gonna-
Count your F
I'm gonna
carbons.
destroy that-
Or
the-
whatever Rick
the
says.
innocence that- yeah.
Just like rewind. So we can only build on the 3 end. So
Correct.
Why are we going from 3 to 5?
Because you are reading 3 to 5. Because complementary is building to the 5 to 3.
So it's
Correct. Yeah.
So there's multiple genes, are there multiple RBSs?
1,000,000%.
All in the same
All in the same spot, same location, same everything.
Because there are lots of rights.
You have
All right.
the same? So that's can
One
I?
second.
One more question.
I know.
One
No,
more
'cause
question.
we're gonna look at this.
Oh, but it was from the last.
So, DNA has your orientations. I have a 5' end and I have mine
3 prime.
3' end. Look at this. That's the light blue. That is 3'.
prime end.
That lines up complementary to.
5.
Go over there. What does it say? There we go. So the nucleotides are coming in and binding here. So the next one's going to bind to that. So I am copying the top strand. I am duplicating that strand. That is what it's going to turn into by binding to the bottom strand. What side do you always build on?
3.
3'. That is it. Always build on
3 prime end.
until I tell you otherwise. Kidding,
In
kidding,
the exam.
I'm not kidding. Nucleotides are, in this case, always being added to the 3' end. We're moving in this direction. Therefore, tell me, where is the promoter sequence? This direction or that direction? To the left. We're over here. Because my +1 is right
There. No.
So the TATA and ATAs are over here.
Add and
Okay.
tab boxes over there on the left. Okay.
Let's see if we believe him. Do you have these ones? Yeah.
Okay.
I'm not sure about that one.
Oh, that's it.
Exactly 50-50.
What?
Oh, now it's 49-51%. Okay. Oh, back to 50-50. Thank you for doing it. I like it. Half, you were right. Figure it out.
It goes off to the left.
To the left. To the left. To the left. To the what?
To the right.
Oh,
Why?
it's off to the right.
It's to the right because the 5 prime of
5
RNA
to
is at the
3,
right.
so it has to be
The
to
5
the
what?
right.
5
What are we
prime-
going to get Samita to say?
F.
The F word. We're going to get her to do it, okay? So we're going to get her to do it by counting the carbons. By not counting the carbons, you're going to get her to say it, okay? Count your damn carbons right now.
Oh.
What direction am I building? Where am I building? This side or this side?
Left side.
Left side. We're building on the left. We're going in that direction. Therefore, where is my +1?
To the right.
Right here. Where is my -10?
That way.
Where is my -35?
Off to the right.
So we're going in that
Left.
direction.
Wait, we're going to the left?
Because the 3 prime is on the right for DNA, which means the 5 prime is on the right for the RNA.
So the answer here would
Promoters
be
on DNA.
To the right.
to the right because the promoter. They're asking about the promoter.
We build 3 to
The
5.
promoter is on the 5 prime end of the RNA.
Okay, that's a good one.
Where is the promoter? Is the promoter on DNA or RNA? DNA. It is telling RNA where to be built. It promotes RNA being built. So the promoter is over here to the right.
Oh.
Okay, so we were wrong.
God, all this up and down, back and forth.
Ah, good question. What carbons am I counting? As a group. Which carbon?
Can I get
1, 2, 3, 4, 5.
That is my 5 prime end. What side can I build on?
3 prime.
Can I get a picture
There it
of your
is.
2 last?
Because what am I building?
3 prime.
What am I building? You're building nucleic acids. What bonds are you assembling? Phosphodiester bonds are what you are assembling by building nucleic acids.